Okay, part 4. How are animals used in biomedical research? The way that they are primarily used is as causal analogical models. Causal analogical models assume several things. Number 1, the first condition that must be met in order for a thing to be considered a CAM is that X, the model, is similar to Y, the object being modeled, in respects A, B, C, D, and E. Now, we know that X, the model, also has additional property F. So, while F has not been observed directly in Y, we reason causally by analogy that Y also has property F. And I'll give you one just simple example. Guinea pigs and humans have a lot in common. So, if you give a drug to a guinea pig and it kills the guinea pig, you might want to think twice about giving it to the human, right? Make sense? Unfortunately, penicillin kills guinea pigs. And that's what's wrong with the theory. Animal models suffer from what's called causal functional asymmetry. What we assume when we do an experiment on an animal is that the same function in species has the same causal mechanism. But we know this is not always the case. For example, causal functional asymmetry notes that the same function can be derived by different evolutionary pathways. For example, birds ventilate through an entirely different causal mechanism, evolutionarily, than humans, but they accomplish the same purpose. And there are many other examples of this. So causal functional asymmetry has major implications for extrapolating data between species. Now, CAMs must satisfy 2 further conditions. The common properties that the model and thing being modeled share, A through E, must be causal properties. You can't just say the guinea pig has a heart, humans have a heart. The properties that you're comparing have to be causally related to what you're studying, what outcome you're considering. And number 2, the properties, the common properties must be causal properties, which, number 2, are causally connected with the property F that we wish to project. So specifically, F should stand as the cause or effect of A through E in the model. And this leads us to systemic disanalogies. Complex systems, like rats and humans and mice, are by definition going to be different in very subtle and unknown ways. We do not know everything there is to know about Drosophila. We certainly don't know everything there is to know about Homo sapiens. And we know for a fact that the same exposure often causes very different reactions in different species. So for a CAM, or an animal model, to be predictive, there should be no causally relevant disanalogies between the systems. And this is just flat out impossible until you know everything there is to know about each system. You can usually find an animal that will mimic a human response. Mice respond pretty much the same way to penicillin that humans do. The problem is you can't have that knowledge except in retrospect. Okay, part 5. This is the last part. So far, the discussion has been somewhat abstract. I'm sure I've explained complexity theory to you such that you now can all go home with a vast understanding of it, but perhaps my skills are not quite that good. But this slide hopefully illustrates in a more tangible way the problems of complexity and hence of extrapolation. Of 10 medications that were withdrawn from the U.S. market between 1998 and 2001, 8 were withdrawn because of side effects that occurred almost exclusively in women. Okay? Men could take the drug and by and large do well. Women took the drug and by and large died or wished they had. Now, men and women are obviously similar in terms of evolutionary biology and gene regulation, but they responded very differently to these drugs. Similarly, a study in Science revealed that one strain of mouse could have a gene removed without obvious adverse effects, while another strain would die without that same gene. Now, a strain of mouse is like a breed of dog. Okay, that's basically what you're talking about. The anti-cancer medication, Iressa, was thought to be ineffective, but upon further analysis, it was found to be very effective for people with a specific genetic mutation. Now, today, because of pharmacogenetics and other advances, we are on the verge of what is called personalized medicine. And what this means is that drugs and treatments can be tailor-made for you as an individual. This, again, illustrates how very small differences between complex systems, in this case humans, can result in profound differences in disease and drug response. If men cannot predict the effects of a drug for women, and one strain of mouse cannot predict what will happen to another if a gene is removed, perhaps we are studying organisms at the level of organization or complexity that defines not only one species from another, but actually defines one individual from another. Evolution, complexity theory, and molecular biology predict that animal testing should not be an effective means of conducting biomedical research. And most importantly, empirical data supports this. Most physicians will tell you that they do not even look at the results from animal-based experiments because it does them no good in their clinical practice of medicine. Now, since the early days of Newtonian physics, important implications for causal determinism theory in biological science have come to light. And this has mainly happened because we've united Darwinian evolution with our knowledge of DNA, genetics, genomics, evo-devo, complexity theory, and so forth. First of all, we know that animals are examples of complex systems, and that therefore small differences between them can lead to changes in the system as a whole in a non-linear fashion. An example of this would be the fact that most of the time, identical twins do not suffer from the same severe diseases, such as cancer or multiple sclerosis, and obviously only a very small difference accounts for this, because we're talking about identical twins. Gene expression has been studied in numerous animals, and the same genes found to express very differently depending on the species, as we saw in that slide. Again, this is because very small changes between initial states in complex systems can propagate into extreme variations. Number two, the level of examination has changed since the 19th and early 20th century. Our examination of living systems has become increasingly fine-grained. We have found that subtle differences between organisms tend to outweigh gross similarities as explanations for biological activity. Science could and did use animals to shed light on shared functions, such as the basic function of the liver or pancreas and so forth. But today we're studying drug response and disease at the level that defines not only a species, but most of the time actually defines the individual. Now, the intact systems argument has historically been the animal modeler's main argument, and it goes something like this. We must test on animals because no experimental system, be it in vitro, in silico, mathematical modeling, and so forth, can predict what a drug will do to the intact living human system. That's basically the argument. Now, ironically, it's the fact that each intact living system is a different complex system that invalidates the use of animal models. Complex systems, as we have seen, are more than the sum of their parts, and different complex systems respond differently to the same stimuli. The implicit claim in the intact systems argument that humans and other animals are the same biochemical animal just dressed up differently simply is not true. Yes, we all have mitochondria, we have ATP, we're made of cells, and so on and so forth, but that is not the question that we're addressing today. Today we're addressing the question of drugs and disease. When we humans were ignorant of the function of the lung, this reductionistic approach vis-a-vis Newtonian physics worked very well. But the level of our examination has so greatly advanced that interspecies extrapolation is no longer useful. And as I said, it is irrelevant to point to observed similarities in genetic makeup between species, since the reasons for the differences are in the regulation of these conserved genes and the resultant interaction between them, not in the genes themselves. As I said, it is as though all species have a common genetic keyboard on which different phenotypic concertos are being played. What matters is not the similarity with respect to the keyboard, but the differences with respect to the order and timing of the pressing of the keys. Studying Mozart's keyboard could not have predicted Ray Charles. The piano itself does not predict the noise or music that emanates from it. Though genes are causal, as the PLOS Biology Journal said, they alone do not determine outcome. Early on, medical research was almost solely the domain of the experimental physiologist. But today, medical research, even the theory supporting it, is multidisciplinary. Physicists and mathematicians are involved vis-a-vis complex system analysis. Evolutionary biologists, molecular biologists, mathematicians, computer scientists, physicists, and many, many others all play different but vital roles. It is incumbent upon all involved to understand the implications of knowledge gained from other fields. And that means that researchers should abandon modes of inquiry based on unexamined assumptions from the 16th, 17th, and 18th centuries. Historically, medicine has been practiced based on statistics. If you suffered from high blood pressure, and research showed 98% of people with high blood pressure responded to medication X, then you wanted medication X. Now, you might be among the very small minority that responded better to medication Q. But because we had no way to determine that, I would prescribe for you medication X. Today we are on the verge, indeed we are experiencing personalized medicine. That is drugs designed, tailor-made for you. Not me, not your mother, not your sister. So, in light of that, considering the implications of personalized medicine, basing treatments on the response of a different species is like using phrenology to study mental illness or trephination to cure malaria. It simply isn't going to be effective. So, finally we come to the harm to humans from studying animals. Now, one question I'm often asked is, why do we do animal experiments? So what if we waste a little money? Big deal. Well, I grant you, as an American taxpayer, I'm accustomed to the government wasting my money. But when we experiment on animals and we extrapolate those results to humans, humans die. And I have a problem with that. And these are just some of the examples. Smoking was thought to be non-carcinogenic for years because we cannot reproduce lung cancer in dogs. High cholesterol diets were recommended. Indeed, we were giving high cholesterol, high fat diets to patients who had just suffered heart attacks. Asbestos and many other environmental-induced cancers were given to the general public without any hesitation because of studies done in animals. And again, we can go on and on and on down the list. To put it in a little bit more modern context, this is a study from Science in 1997. And the National Cancer Institute tested 12 drugs that were currently being used successfully to treat cancer in humans. And they studied mice that were called nude mice or xenograft mice. And these mice were growing the same cancers that these drugs were used to treat. And indeed, they were human cancers because they took a bit of the human cancer, implanted it into the mouse would grow it, and then the scientists would give the mouse the appropriate drug. Well, what this study showed was that 30 out of 48 times, the drugs were ineffective. Okay? So 63% of the time, even though we knew that these drugs were effective in treating human cancer, they were ineffective in treating mouse cancer. Now, this same article goes on to suggest that perhaps we have lost cures for cancer because they were ineffective in mice. Okay, that's the National Cancer Institute saying that. That's not me. Drug testing has come under fire for many decades. Here's just 4 studies, and there's about 10 to 20 studies out there that have looked at drug testing just like these have. I picked these because they really show the range. These studies show that animal models failed to predict drug toxicity between 43 and 95% of the time. And you'll see they failed to predict 43%, 67%, 83%, 95%. I don't care how you do the statistics, it doesn't work. Okay, predicting toxicity based on animal models is a failed paradigm. So why should we abandon it? Well, very simply because it doesn't work. Okay? I don't care what alternatives you have. We don't do bloodletting today. We don't do phlebotomy to treat malaria. And the reason we don't do that is because it just doesn't work. All right? So regardless of all the other reasons, animal experimentation using animals as cams to study human disease and test drugs should be abandoned. It diverts money from viable research modalities. We have better options. It misleads researchers, and so on and so on and so on. Okay, now does this slide make a little bit more sense since you saw it for the first time? I'm running out of time, so I'm going to go ahead and go to the next slide. This is a very complicated subject, okay? I essentially have read hundreds of books to get the knowledge that I need to give even this very short, much too fast presentation. And if you really want to understand why the animal model is not a scientifically valid paradigm, you have to know about gene networks, you have to know about gene regulation, you have to know about complexity theory, you have to know about Evo-Devo, and these are just some of the books that we recommend that you read, and we have a much longer list on our website. Let me close with this slide. What a difference a little DNA makes. Thank you very much.
Thank you, Dr. Geek. I will now call on Andrew Skolnick, but it will take a couple of minutes. I think he needs to set up his PowerPoint.
I think it's set up.
Set up? Okay. Before I start the clock, have a couple of minutes.